Yeah, thank you so much, Tom. Incredible. You're an entertainer, an organizer. It's fantastic. I have a bad voice because of too much screaming last night. But I enjoyed to be in Copenhagen because Copenhagen was quiet last night in contrast to Germany. Thank you so much for the invitation. It's fantastic to be here. And maybe 2 words before I start the talk. Thanks to Anton, to Morten, to Tom for getting us here together. It's really a wonderful, wonderful moment, a wonderful possibility. And you are the best organizer ever. I didn't get so many mails from any organizer in my career as from you, including the newest weather forecasts and what to wear and what not to bring and what to bring. Fantastic. Thank you so much. I think this was just wonderful. And the second is this talk is certainly not my standard scientific talk, as you can imagine. When Tom asked me to do that, it's a great pleasure and an honor to do that. But I tried now to sum up, to wrap up, and also to bring you to points of thinking and new directions. And this is, I think, the whole point of this meeting. Making science as friends, making whole genome science as friends, but also think about the new avenues. And it's a pleasure, and I will be with you for the next 3 days. I'm very much looking forward to talking to you. Now, let's see if this works. Yeah. So Tom already showed the first one who was rethinking our nature. And that is, of course, was Charles Darwin. And he was the first who said that humans are just part of nature and not something very, very special. And if you read his book, I'm not sure how many of you really have read it, but if you have it, it's a wonderful. In the very last end, there is a chapter which is called the Entangled Bank chapter. And when you read that chapter, I have highlighted it here, then Darwin says it's interesting to contemplate on entangled banks and to reflect that these elaborately constructed forms, so different from each other and dependent on each other in so complex a manner, have all been produced by laws acting around us, organisms depending on each other. For me, one of several turning points in my career was the fact I always thought that I'm working on an ancient animal and I can maybe help to uncode the laws which are regulating development from the very beginning of multicellular life. But I was on a conference and Andy Knoll at that time talked then to me. He's one of the famous paleontologists of this world. And said, well, Thomas, it's all very nice what you talk about, but look about here, it's 560 million years. This is your organism, but 3.5 billion years. The world was colonized and inhabited by microbes in all kinds of different manners, and they have invented our language, how we talk to each other in biochemical terms. They have to manage how to live together with very different genotypes, and so that was the origin of everything. And of course, at the end came the origin of the eukaryotic cell. From all that, it's very clear, after all these sequencing events, etc., that we are dependent on microbes, and I don't have to tell that here to the people. And we came to the conclusion that organisms cannot be separated from microbes anymore, and we have to expand a bit the view of the host. And this is what biology and medicine was focusing on about the last 150 years, the host exclusively. But this is not leading to fundamental correct answers. You only get to an answer if you accept that organisms are holobionts or meta-organisms, as we told them. We still have these two terms, but what we mean is we all and any organism on this earth is a multi-organismic entity consisting of different kind of prokaryotes, of viruses, of symbionts, small ones, of fungi, we shouldn't forget, etc., etc. And it's very clear that there is, we always phrase it when we write a review or something carefully, but there is literally no germ-free, bacteria-free organism on this earth for a very good reason. It's the animal condition, and you can also say it's the plant condition to have microbes. Otherwise, and I will show you that, they will not function. It's ignored these days, but it started early time on when people, I mean, germ-free animals are around after the First World War. Actually, the lady who invented them, she's still living in Prague, and there were germ-free pigs for some reasons. But for these colleagues, it was very clear that the animals are not normal. And if you open up a germ-free mouse, and I brought it to you because nobody of you probably has ever done that, because you only look in the computer and see your sequences. But if you open up a mouse which is kept germ-free, the anatomy is very different in many different aspects, organs, organ size, but also the physiology, and the very same is true in plants. And if you take away the microbes, as we all do now, because we keep germ-free animals or germ-free plants, then of course we know that they are not normal. And I could also say they are sick, but it's of course a wonderful new facility to study the functional roles involved. Maybe, I don't know who did it, the publisher who made the revolution out of the Holobiont, but of course it is a revolution, because when we start, I mean, Lynn Margulis, the origin of the term is a bit controversial, and there are some persons who claim it to have done it, but accepting that's probably Lynn Margulis who was the first one who termed the term Holobiont. Nothing happened. As all of you know who follow Lynn's fate in science, she was ignored during her lifetime, more or less, completely. But she did so many things right and a few things wrong. And so that was when she introduced this idea of this holobiont. But look at that in terms of publications now. Nothing happened until very recently, when then it's now a boost in the literature. And this is just a recent snapshot from one of them. And so we contributed to that in many different ways. And the field is still exploding, and I'm so happy to see so many young people here, because it's your future, it's your field, what you can develop in many different aspects. When we came to this thinking of, I mean, you know, if you would have asked me 25 years ago, I'm working now mostly on the interaction between microbes and an ancient multicellular organism, I probably would have carefully listened to you, but it was not in my mind. You're driven by questions and by new challenges. And if you get to a point, which I show you in a minute, then you have to accept, okay, that's now a turning point. Either I go there or I go on standard manner, and then it's getting boring, and I don't want to be bored in my life. To summarize these new findings, and there was a theory by Eugen Rosenberg and his wife Ilana Silber-Rosenberg, the so-called hologenome theory of evolution, which brought that very nice in a nutshell. Unfortunately, oops, I'm getting hooked up with my cables. Unfortunately, they became a bit in a controversial time. It was early times, and people, you know, didn't believe, do we need another evolutionary theory? No, we do not. But the life is getting more complex. And so this is the hologenome theory of evolution, which says that there is a genome of the host, maybe with a bit of mitochondria, but there's also the many genomes of the thousands and probably millions of microbes. And Robert Brugger and Seth Bodenstein, who is here in the audience, I asked him at that time, I was editor of a journal, and I asked him to make a kind of, today you would say, a graphical abstract, and I think it makes the point very, very clear. What we have is the stable and hard to change host genome, not to speak about the complexity of the mitochondrial genome or the chloroplast genome. But then we live now, that's the holobiont imperative. We live together with thousands, with millions of different microbes, and some of them, they contribute in terms of metabolites, in terms of features, physiological features, genomic features that contribute to this host genome. So you get much more richer, and you get different because of your microbes. And you may adapt to environments which you couldn't do without your microbes. But this graphical abstract makes another point very nice. The microbiome is much more flexible in terms of being together at a certain spot and with a certain host. And so there are microbes outside of this inner ring of hologenome. But if you choose, you move into a different habitat, or the habitat changes because of anthropogenic reasons, then some of the microbes don't feel well anymore. And so they may disappear, but others come in. And this flexibility, this dynamic, which is not possible with the host genome, that's the power of the microbes. But that's also the power of the holobiont, because it is a complex, multi-organismic entity, and it's still, for me, a complete enigma or maybe a wonder, because all these microbes are completely different genotypes. In terms of phylogeny, they are completely unrelated. But they all somehow manage, and we are in a kind of a homeostatic organism and doing well until we disturb that, and I come back to that at the very end of my talk. So the point of that is that the focus on I'm talking often to human people, but so that is even more important to make the point than to you here, of course. The point is focusing on the host gene alone host genome is completely misleading, because we do not understand then the complexity of the features which the host can do, or the complexity of diseases the host suffers from, and that's what personally drives me in Kiel University. So we have really to move on, and we have to move on in a way which Tom Gilbert and Anton and Morten and all the center here and the team here really made it the point, and this is this holomics. We have to consider now the whole complexity of many different organisms within as holobionts, but also the interaction with them, and then going to the applied hologenomics, because we know that this being together has, in terms of agriculture, in terms of farming, in terms of aquatic organisms, in terms of medical crisis, in terms of plant adaptations, in terms of climate crisis, so many different applications. And we can only approach this complex real life if we do hologenomics. And this integrated approach, which was, I would say, invented here, and we should all be very, very thankful that Tom did that. So giving a bit what we do and how do I integrate that here? I mean, first of all, I wasted all my time with a simple organism, Hydra. This question I get very often: why do you waste your time with Hydra? Don't do something really important. But this organism is a beautiful organism for studying complexity, because the organism is simple, but it is complex enough, and in terms of biochemical language, nearly identical to what even a human being is. And the organism has a clear pattern, has all the genes which are necessary to make it, and it is very well studied at the cellular level. We have contributed to that many, many years ago with stem cells, and we understand the organism pretty well, and it's kind of transparent, small. And then if you do that for 20 years, you think you have understood the organism. But that was a shocking moment in my life when I realized I don't even understand this simple organism. Because first, only by sequencing, of course, we are going an invisible way and discovering, first of all, invisible components. We only could guess that there are microbes, but now we can visualize them. And in the early days, what we had no idea is that the ectodermal epithelial cells produce a mucosa of highly conserved mucins. It's the same mucins as you have in your mucosa in the gut. And in this complex mucosa, which is called glycocalyx, live a very complex microbial community, which we now can visualize and study. And when it comes to that point, you really have to say, do I really want to go into this invisible territory now, or I ignore it because I'm doing what I'm used to? And I decided not to ignore that. I decided not to ignore that because I had a fantastic graduate student at that time, Sebastian Fraune. He's now a full professor at the University of Düsseldorf. And when Sebastian came in the lab, we sequenced different species of Hydra in terms of the microbes, of bacteria. And what Sebastian very early on discovered is that different species of Hydra have very different microbes, and the differences are correlated with the phylogenetic relationship of the host species. So you have a clear kind of a co-evolution or co-diversification between the host species and the microbiome. And so we did it twice because at that time it was very unusual. And actually, reviewers from the first PNAS paper outed themselves during the reviewing process and asked if they could use the data. And it seems that is a general phenomenon. And when you then, as a biologist who loves Hydra, who loves the species, you realize, good Christ, all of this phylogeny here has to be understood in the context of a microbial phylogeny, and the different colors are just the different microbes here. That really made the point where I said, I have to understand now the Holobiont Hydra, or the meta-organism Hydra. So the microbiota is a complex trait that is under genetic control, obviously, and we have understood that much better, always going from an evolutionary point of view, but strongly believing that what was invented in nature 560 million years ago, that probably has for a very good reason if it's still today. And Seth Bodenstein and his group at the time then came and gave a term to that phenomenon and gave the term phylosymbiosis, that in many different species then it became clear that you have here, I show you a few dates on the great apes, by the way, and Seth worked on wasps, so that in many different host species you can really follow that co-diversification or co-evolution. And what that means is there is selection processes going on in microbial community formation. That is, if you are a biologist, an evolutionary biologist, an evolutionary developmental biology, that is then so strong that you really are, you want to understand that. What the hell, why is that so, why are holobionts now the unit of life, if you want to say it so. And this is supported now, and it's getting supported every month in different papers. This is from a Kiel group. Malte Rühlemann, he works with Andre Franke in our center, and they study hominids, so humans and great apes, and so there's a very clear phylogenetic, phylosymbiotic signature in the humans and the great apes and the microbiome. And when you go here and see here to Morten, who is in the audience, he published recently a paper on salmonids, on fish, and a certain microbe. So you can see that now in many different aspects. So microbiota colonize a host in a species-specific manner. I think that is a very important point. It is so important that it now should lead us to rethink a few concepts which we have developed over the years. And I go through these 5 aspects now, starting with the immune system and development, and then the neurosystem. We go to behavior, and I would then end up with a kind of a final points, which are just for discussion for the next 3 days. It started very conventional, and now that's true for all organisms studied so far, by rethinking the immune system, by finding that microbes are important in the immune function of a given host, And the finding was relatively trivial because first of all, we found that these bacteria are there, but then if you take them away and make germ-free animals, that was great. And it was another graduate student, Søren Franzenburg, and when Søren came and said, well, I have germ-free hydra, that is fantastic. And 3 days later, he came then back with a very bad mood and said, Thomas, I'm very sorry, but all our germ-free animals have fungi infection. And we never have fungi in the lab, and never in our cultures. And I said, God hell, too much champagne about celebrating the germ-free animals, but now they are all dead. And they are dead because, so this is the normal animal, but these are now just germ-free animals, and they get, you make them germ-free by antibiotic treatment, so they get infected with fungi. And they get infected because that is the epithelium of the animal, and outside the epithelium are the microbes sitting in a complex community. That is important. Diversity matters, as it matters in ecology. So it must be many different ones, and they interact. We have shown all that. If you colonize with just one, it will not help. But when they interact with each other and they produce something anti-fungicide, and so they protect against the fungi. So it seems that microbes contribute to what we call today colonization resistance against fungi infection. And the same happens with you. If you've been yesterday with the dentist and he said you have a parodontitis and you need an oral antibiotic, and today in the morning you looked in the mirror and you fainted nearly because you had thrush in your mouth, and thrush's candida grows, and candida is a wonderful partner in your oral cavity, but if you remove the bacteria, this fungi has so many niches free and can grow, and then it causes this thrush, and you have a hard time to get rid of that again. So the same principles, nothing makes sense but in sense of evolution. And then we went in the innate immune system, and the innate immune system is very classical, starts with the toll-like receptor, the signal transduction cascade at the end, NF-kappa B, and then antimicrobial peptides. And the antimicrobial peptides at these early days, what is an antimicrobial peptide doing? It's killing. Yes, it is killing. But we know now that any organism has a huge diversity of antimicrobial peptides, thousands of them, large families which are encoding many, many different ones. Why the hell? It's costly to make so many antimicrobial peptides. We know that the antimicrobial peptides are much more regulators, and they regulate the composition of the natural microbiome, and that is true in all organisms. And there's beautiful work from Bruno Lemaitre's lab in Drosophila recently done, which clearly shows that, and Bruno took all the pain to do genetics by knocking out single Drosophila antimicrobial peptides and looking what happens then to the beneficial microbiome in Drosophila, etc., etc. So antimicrobial peptides are the interactors between the host and the symbiotic prokaryotes. And I come back to that. They are still killing. But they're not killing just for killing. They kill because they keep the homeostasis. They keep the species-specific microbiome. And if you disturb that, we did knockouts with antimicrobial peptides, then the animals get a wrong symbiotic microbiome, and that makes them less fit than their control parts. I'm a developmental biologist by training, and so it was very obvious to get intrigued by questions which affect development and life history decisions. And they were wonderful and important, which actually brought the field forward, findings from colleagues from different fields. It probably started old with the legumes and the plants and the microsymbionts, nitrogen reduction, etc. But then came the beautiful work by Margaret McFall and Ned Ruby, where they showed that a single bacterium, Vibrio fischeri, can induce the light organ in the squid. And we have then a beautiful early work also in the mammalian systems, where we see that development. And I showed you this opened up germ-free mouse, that development of the anatomy, morphology, physiology of a mammal is also to some extent dependent on the microbes. We do not understand yet how. This is one of the open questions yet, but they do. And if they do, then you may think that this can be applied by maybe optimizing tissue differentiation and tissue repair. And there's a beautiful review in Cell just came out a few weeks ago. And, Monica Medina, who will be with me doing the discussion later, had a wonderful paper where she showed that Cassiopeia, one of these jellyfishes, there's microbes which are important for life history decision because the larva has to make a decision, I have to settle here. And if they settle here, they never can move again. And that's true for many marine organisms. So that's the one lifetime decision. If you decide to settle somewhere, that's it. If it's the wrong spot, you're gone. So, and in this case, many of these microbes play an important role. Hydra is maybe the oldest model in developmental biology invented by in the 18th century already, but was used as a model to understand basic developmental processes, and we have started to molecularize that, and so Hydra has a clearly pattern. There's a top part, and so it's very different. You're shown by transgenic cells. But I show you now unpublished data, and don't worry, tweet and Twitter and whatever you can do, I don't worry, nobody can steal anything from that, but I think it's fun to present to an audience like you as a not published work. The work is 6 years in progress now, thanks to a fantastic graduate student postdoc now. And it started with a very weird observation. To read, to learn new things, read old books by my late mentor, Pierre Tardon from Switzerland. It's true. And there is I would say a beautiful paper. Some people say it's a very weird paper, published in Science in 1982 by two Israeli colleagues, Menachem Rahat and a colleague. And what they published in Science is cultivation of bacteria-free hydra, a missing budding factor in non-symbiotic hydra. If you're in this field, you say, wow, you take away the bacteria and they cannot do one of the most essential life history decisions, and they propagate mostly asexual by budding. So in absence of bacteria, they cannot do that anymore. And this is a highly orchestrated patterning process. The bud comes at a very certain position of the polyp, and then it makes a new polyp. Do bacteria have such a strong influence on that? And many people, including us and my early team, tried to find or to repeat these experiments. We couldn't repeat that successfully, and the factor has never been characterized. Until Jinru He came in the lab, and it needs certain persons for challenging projects. And Jinru managed to keep animals germ-free for more than 3 years, and that means 365 days of taking care of them, sterile feeding, sterile culturing, etc. And what he observed is in long-term germ-free animals, bacteria-free animals, that indeed they change. First of all, they stop budding, and then they get larger, and they get fatter, and then they get weird, and they have a patterning problem. And with a molecular probe, you can see that they have foot-like structures along the whole body column. If you follow the budding pattern, you see that the budding pattern ceases with time. The animals stop budding. But if you add back microbes, they start to burst, as if something is accumulating which inhibits budding. But then if you add the microbes back, buds come and head structures come all along the body column. And when I saw that some years ago, this organism, actually we showed it to referees. We had no paper, but I needed money. So I just showed the dishes with the mutants to the referees, and they were convinced and gave us the money. And I promise the paper comes out now this summer. Thanks. This is a good referee, right? Who accepts that. So it's reversible. This non-budding, and you can do that by adding back the microbes. and make a very long story and a beautiful story very short, but then we did single-cell RNA-seq, and we made an atlas. You have seen these atlases for many different organisms. Tom is an expert in making this kind of atlases, and what you can see is that in non-budding animals, this is now gene expression in stem cells in the animals, in controls, in different controls, short-term germ-free, but long-term germ-free, which are non-budding. And what you can see is that the gene expression pattern in stem cells changes dramatically. And which you can reduce then back to if you are back in normal animals. And then you can do a trajectory analysis with this new type of generation of data. You can do all kind of bioinformatic analysis, and you can now follow the cell when the cell is in a stem cell compartment and it moves out of the stem cell compartment and it is in a phase of transition, and then it's a terminally differentiated, probably a head-specific or a foot-specific stem cell, and or a differentiated cell. And when you look up, this is the controls, and when we do this trajectory analysis in non-budding animals, you see that many of these genes, and these are all these many gray bars here, many of these genes, this brachyury here is not present in a non-budding or not expressed, but it is present here, and there are all the Wnt genes which we knew are important, but even more important and eye-opening, because we took germ-free animals, we learned about the basic rules of pattern formation, that upstream of Wnt are specific genes which are controlling the downstream Wnt-regulated machinery, and upstream genes are among these genes here. They are already differentially regulated in the stem cell compartment in the absence of microbes before the cell goes in the transition. And we have now knockdown mutants of these genes, and the genes are expressed in the right compartments here. So for me, that's a lesson, and please tell that to all your colleagues who are ignoring the microbes, that you may be interested in the microbes and how they interact with the host, but you also learn something about the system per se, and we never would have discovered these genes if we wouldn't have done this type of experiment, because we are very much hooked up in the Wnt language, but everything to everybody in the community was clear there must be something upstream. The absence of microbes has told us what is upstream of the Wnt, and our current model says that under normal situations the stem cell has to make a decision, and it goes into a head-like area and a foot-like area. But if the microbes are not there, something inhibits that the cell rolls down the Waddington landscape in this and this direction, but it only goes in the foots, and that's what we see here in these animals here. Developmental biology needs a more holistic view, recognizing that animal development may never have been autonomous, but rather requires transient or persistent interactions with the microbial world. Our centuries-old host-centric view will inevitably fade in development as the interplay between an animal host and a multitude of microbial prokaryotes will emerge as a rule. I can give as many talks as I can and traveling around the world as much as I can travel, but then there come reviews on the revolutions in developmental biology, rebranding developmental biology, and microbial symbiosis is not mentioned a single time. So it means keep on going traveling. Next chapter, rethinking the neuronal processes. That's getting really fascinating now, because we enter now what you are used to think about the brain-gut axis. But in an organism which has invented the nervous system. The nervous system, the origin of the nervous system goes back of course, to the pre-bilaterians, but then to the cnidarians. There is no nerve cells in the placozones or in the porifera. This is a controversial, interesting group of animals here, which I ignore. And we are just focusing here on this sister group to the bilaterians. Means all what we learn here has something to say over there, means to us. The Nervous System of Hydra, this is a book which is still on the market, and which is terrible, because it says primitive nervous system, and it shows this super simplified thing. And that's what kids and students learn in their basic lectures. It's wrong. The nervous system of Hydra is beautiful. It has invented the neural language, which is now 560 million years old, and it's very successful. And your neurons use the very same grammar than these neurons use. It's crucial for understanding our nervous system. It lacks a centralized nervous system. It's morphologic simple. And a lot of work in the last few years has shown that this Hydra nervous system has at least 7 different subpopulations. It has a CNS, central nervous system-like, and enteric nervous system-like networks. Many, many neuropeptides. Some of them are antimicrobial peptides. Neurons contribute to that. And it's involved in controlling resident microbes. That's why I put that chapter here in this audience. If you are looking into this, there should be now running all the panels, and they don't. Maybe we should. No.
It is?
Yeah, no. The most beautiful animal is not pumping. So, all organisms in this earth have the ability to contract. Sometimes I call it the pulse of life. And if you disturb that for many different reasons, you kill the animals. And so these are Hydroctinia, these are jellyfish, this is a worm here, this is a mouse gut isolated in an aquarium. This is a human small intestine I got from my friend Stefan Schreiber, actually from a Parkinson patient because it goes backwards and forwards. So if this is disturbed, you have trouble. And this Parkinson person has trouble because it's arrhythmic, and it goes in both directions, and if it stops completely, you have obstipation, and that's a lethal disease. I show you that because when we came to this nervous system issue, we first studied the beauty of these different organisms and found that all of them can do that, but who regulates them? So another very nice graduate student, Andrea Muggillo-Rincon, now a postdoc at the Max Planck Institute, she measured that contractions here, not only the amplitudes, but also the periodicity and the regularity of these contractions. And she found something really astonishing and was not taken up in the scientific community so well as I have wished for, because if we remove the microbes, then A, the number of contractions, and B, the periodicity, which is not shown here, the periodicity is changed drastically, and you can rescue that by giving back the full set of bacteria, but not, you cannot rescue, you cannot rescue that if you only give back one single bacterium. And so again, the diversity matters and the full complex really matters. And so the microbes, for some reason, have something to do with these contractions. And many studies later, we found out that this reason is actually pacemaker cells. And so I give you only a very brief summary. This is a very nice paper in PNAS with Alexander Klimovich, a senior postdoc in the lab. And so what we have drawn in this paper is a kind of an archetypical pacemaker cell, which is the same in your gut or in a mouse gut, and also actually in the C. elegans worm than in Hydra. And it has 2 sets of receptors. It's innate receptors, and which are accepting signals from the bacteria, but they also have signals and signaling and antimicrobial peptides which are going to the microbiome. That is in addition to all the classical pacemaker biochemistry, the biochemistry which links the pacemaker cell to the microbiome. And that's true from Hydra to man. So from that we come to the conclusion, since we are working at the very origin of the nervous system, that it seems that in addition to sensory input and to motor output, the central function of the nervous system from the very beginning when it was invented was interaction with the microbiome. That's an interesting case because you have here an holobiont which invents a new organ. There were the animals before them had no nervous system. Now you invent a nervous system, but you never invent an organ which has nothing to do with this multi-organismic entity. You invent the new cells and the system immediately integrated into complexity. That's what these experiments showed to me. It's getting even more complex. because when we look into the output of these neuronal systems, and this is behavior, or at the beginning, simple reflexes. And I will show you that even simple reflexes are heavily dependent on a diverse microbiome. And what we study here is the feeding reflex of hydra. And when you feed them, it's lovely to see. And everybody has done that maybe at home, or we send the animals to school kids, then you can see the animals catch them. But then they're opening the mouth, and then they engulf the prey. And that feeding behavior is very canonically, and it was actually described already 50, 60 years ago, and it follows certain aspects of tentacle wavement, and at the end, the mouth opens, and then the prey goes in. And so we ask two questions. One is, what are the neurons which are involved in this mouth opening? And B, what is the microbial contribution? Because we had the observation that germ-free animals, as germ-free mice, don't eat that well as normal animals. And there can be many reasons, but if they cannot open the mouth, then it's not easy to get enough food. So this is the really beautiful work of another graduate student, Christoph Gietz. Christoph moved 2 months ago, I think, to the Francis Crick in London as a postdoc. And what Christoph did, I mean, you know, when you do these experiments, I know that you all look at that and say, it's very nice. But you have to put hard work into that. And it's 10 years of molecularization. And now came Christoph, and actually 2 years of his PhD study was developing new methods. And what we did is we brought the calcium imaging constructs into the animals so that we can quantitatively quantify the neural activity. But then automatic imaging and behavior, of course, so that we can follow that. You see here an animal which is opening the mouth. We don't need to feed them. We give them a molecule, GSH, and then so we can then automatically monitor this behavior. And last but not least, very important for the field, Christoph managed to ablate cells in vivo in the cell in the subtype-specific manner. And so we have a subtype-specific promoters, means neuronal subtypes, so we can create a tissue which lacks this population or lacks this population. The animals are still there, and they are fine. So we can study the complexity of the nervous system in the absence of some of the neuropopulations to get an indication what are they really doing. And so to answering the first part of my question, which neurons are involved, here's the food coming in, and then the first cell population which really fires in calcium imaging is the N6 population, and the second population which then immediately follows is the N4 population, which is a network along in the body column. This is canonic, and the crucial experiment was when we took away these nerve cells, the nerve 6 or the nerve 4, then mouth opening was not possible anymore. So we have uncovered and identified and causally shown that these two populations are really involved in the mouth opening part of the feeding behavior. But the fun part came because, of course, we had the observation that in the germ-free animals, feeding behavior was less effective than normal behavior. And then Christoph made this, at the moment, very confusing observation that when we monocolonized the polyp with one bacterium only, Curvibacter, the animals couldn't open the mouth anymore. And Curvibacter is a part of the normal microbiome. All the other bacteria, we have all the bacteria of Hydra in culture, all the other bacteria. So the germ-free are a bit less effective, but monocolonized with Curvibacter, they could not open the mouth anymore. This is where the Curvibacter sits. We have a transgenic bacteria where we can localize their normal feature. Then the next question was, why the hell? What is going on? And so then Christoph found out that Curvibacter produces glutamate. And if we add pure glutamate, again, no mouth opening. So the bacteria produce glutamate, and we have a glutamate receptor on the N6 neurons, and then this leads to this disturbed eating behavior, and the pure glutamate does the same thing. The confusing thing, if you're still with me, is that Corpibacter is part of the normal microbiome. So why the hell does Corpibacter, in the presence of the other guys, not show this defect? And we could show that if we give back the bacteria in addition to Corpibacter, then we could rescue that behavior. And the answer of a long, long story, again, 3 years of work, is that there's another bacterium in this microbiome, which is called Undibacter. And Undibacter desperately looks for glutamate in the microbial community produced by Corpibacter. And it cannot, because it cannot make glutamate by itself. And then Corpibacter provides the glutamate, and Undibacter takes it up. That's a normal life in this independent microbial world. They don't care about the host. They live their own life. But if you disturb it and you remove Undibacter, then this glutamate is accumulating and accumulating, and it does bad things. So we know that a normal microbiome has no problem. But dysbiosis, and that is a word of these times now, because we all live in a time where we completely disturb the microbiome in many different aspects because of our lifestyle, then it inhibits this response. So summing up this nerve cell part, multiple populations and complex behaviors, pacemaker system. By the way, the system evolved before the mesoderm, so that means the immune cells are in the ectoderm, and that's why nervous systems are immune systems, and it co-evolved with the specific microbiome. A model is a lie that helps you to understand the truth. I think this is a very good description of what a model really is. That also tells you how good a model really can be. And what I've shown you with this model is that we have developmental impact of the microbes, that we have immune impact of the microbes, and that even microbes are part of ancestral neuronal circuits. So we certainly have to think beyond the host when we want to understand development, neurobiology, or any other immune biology or any other aspects. Before I come to the end, I thought that I confront you with 9 ideas, topics to discuss. Maybe you find them irrelevant, but which I think could be future frontiers. At least we have not understood them yet. And I will go with one slide through all of these different frontiers: the ecosystem, the missing microbes, microbial biogeography, the epigenome, biodiversity. Rewilding is getting a hot topic, the built environment, and a new language, and the concept of self. And again, this is just a very nice review. It's called the Eco Holobiont. What this picture should show you is we need to get to a holistic view of ecosystems, terrestrial and aquatic, and probably both together. And because we talk about holobionts, and we will hear many lovely talks of holobionts the next 2 days, but in the essence, all of that has to be understood, and that's probably also the essence of this modern term of One Health. Remember what Charles Darwin said: dependent on each other in so complex a manner. The second issue, which personally drives me and keeps going, is, of course, that we do a lot of bad things to the microbes in our Anthropocenic time. And Marty Blaser, Missing Microbes, his book has probably everybody on his bookshelf here. But Itzik Mizrahi, who is in the audience, has a beautiful paper just came out where he shows that the decrease of cellulose degrading gut bacteria in the industrial societies and not in the rural societies. And we could, of course, add there all of the many the findings of Gloria Dominguez-Bello and many others here. So holobionts are at risk, and that's, I think, an important point to look at. Another issue which seems to me not yet understood well is what we call microbial biogeography. When I took this term first in the mouth in a meeting, my goodness, I was thrown with stones. Don't say biogeography. This is a fixed term which is used by whatever geologists or whatever. I talk about microbial biogeography, and what I mean is that your body and any other body, that's the microbial biogeography in hydro. There are some guys which are sitting up and some are going in the down along the body column. Why and how? The host selects very specific. You have very specific microbes in the intestinal niche. How do they, which molecules are keeping them there? And why are they sitting exactly there? And we have, there's a beautiful review, I think I have added here, from Carolina Tropini from Vancouver, which shows you that if you disturb that biogeography, bringing microbes to the wrong places, that's not good for your health. So why the hell is that maintained? No idea. The epigenome. Everybody talks about epigenetics. And yes, we know epigenetics is the way how to communicate with the natural environment and with stress factors and environmental factors. But how does it really work? It's a beautiful work coming out from here, or a beautiful review here, just published a few days ago, I guess, which shows you that the epigenome and the epigenotype is the host epigenome is interacting with the microbe, and it's not understood which kind of microbial molecules are interacting with which kind of epigenetic enzyme machinery. But it is important because the host is stabilized by an epigenetic signature, and changing the signature has dramatic consequences. And if microbes contribute to stabilizing that, good hell, if the microbes are not there anymore, this may have a lot of impact on the host phenotype. I'm visiting a lot of zoological conferences, and biodiversity is a major issue. But I can tell you, and probably you have experienced the same thing, you may sit 3 days in a zoological symposium or a workshop on biodiversity, and you see all this beautiful work. But then after 3 days, you realize, my goodness, they didn't talk a single time about microbes. And biodiversity loss is a major issue these days, yes. And species are disappearing, and insect species are disappearing, and frog species are disappearing. But they are not disappearing just of nothing, because then a population decreases and disappears. The trouble is, because of the environment, the changed environmental factors, and the dysbiosis in a single holobiont individual, whatever an individual is, causes then the loss of the species population in the long run. So we have to bring that together, the micro biodiversity researchers and the macro biodiversity researchers, and there are by Gabriele Berg and others are nice reviews on its way. If you think about that diversity loss and microbial loss, and yes, our lifestyle and environmental factors contribute to that, you may think of counteracting that. And I think the trigger paper was from Barbara Reichermann, where she showed that true wild-type microbiome in the mouse is so much more powerful to keep the animal fit than the normal laboratory microbiome. And the term came up, rewilding. Rewilding means that you bring in your host the true diverse microbiome, hoping that this microbiome can bring back features which are counteracting complex diseases or making the animals more resilient against environmental factors. And that brings me to thinking. I'm a member of a Canadian think tank, and at one breakfast we were thinking together, and actually I had a sabbatical in Berlin, and I met architects and leading architects of this time, and Beatrice Colomina and Mark Wigley and Forrest Meggers, and so we were drinking and having a glass of red wine in Berlin together and thought about the same issues. They are architects and designers and leading schools in Princeton and in Columbia. And so we thought, good Christ, what are our modern buildings? Central heating, central ventilation, etc., no open windows. What are they doing to us? And thinking back about evolution. And in evolution, the buildings were very different. They were open, they were dynamic, there were flexibility. There could be input and output. This is very human-centric microbiome here, but we spend 12 hours and more in closed environments. And I spent many nights in hotels where I cannot open a window anymore. Thank you, we can do it here. And so this is thinking about what the new generation of architects, that's why we put this paper out. We want that they think about the problem. And we don't want to go back to huts, and we don't want to go back in the jungle. But there are modern ways how to keep a building microbiome friendly. What about our language? People thinking about that, well, microbes have such an important impact. So there's this wonderful work by Albrecht and the symbiosine. And so this is the age of the symbiotic organisms here, which is a kind of a term. But if you think a bit more deep, then you realize that the way how we communicate with each other is actually with a language which was invented when we were only used to think microbes as pathogens. And that was golden and important times in microbiology with Virchow and all the other guys, because, of course, we got rid of all these bad pathogens. But this language is a war language. We get infection and pathogens and antimicrobial peptides. But now the world is very different. We live in the symbiosine, if you want to use this term. So we thought of maybe we should also adapt the language, and maybe instead of infection, colonization, and maybe instead of antimicrobial, maybe a regulating peptide. We don't put out a lexicon which we force you to use, but we want to make you think that the language at the moment doesn't fit anymore to our findings. And last but not least, intellectually and philosophically, of course, fascinating, the question, if that all true, and Tom says it's true, what do I say here? That we are holobionts, multi-organismic. And if you have listened to me the last 40 minutes, I try to persuade you that this is all pretty important interactions. And each single one, if you interrupt, you don't function anymore normally. What does that really mean then? Who are we? Are we humans? This is a book by Beatrice Colomina, by the way. Is the human more than the human? Or what is the self? And we have a paper where we thought about the initial definition of self is because of the immune system. But now I told you that the immune system is heavily supported by non-self microbes. Then you say, maybe, yeah, okay, but I have my the genome, and this is me. But in an earlier paper, we showed that 37% of your genome has bacterial origin because of the history of the human holobiont. And then you say, no, my brain is my brain. But then think about the gut brain. So I don't have answers to that. I spent this morning in the local art gallery here, and I found wonderful the Munch and this Berenskreuz, where this was a time where artists tried to overcome the separation between man and human. And man is part of human. And man is part of nature and not separated from nature. And this is an interesting, if you walk through an art gallery and you go from the 16th century or the 15th century and then walk up, and then there's a critical period of time where Edvard Munch and some paintings which you find here in Copenhagen are then depicting that. I'm at the very end, and I want to summarize and coming back to the founder of the whole field. Tom showed his picture at the very beginning. I showed you the entangled bank citation. And we read the very last sentence from his book, from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. Endless forms, most beautiful. I thank a lot of people which have contributed over the years to this, collaborators, fantastic students, changing students, young students, older students, funding agencies, and I thank all of you for your attention. Thank you very much for coming.
Thank you very much, Thomas. That was wonderful. I'm not going to let people ask questions now because the whole next hour is Thomas and Medina in a discussion where there will be an option to ask questions. So if you'll just give us one moment, we're just going to set up for that.